Synthetic biology is an interdisciplinary branch of science that combines principles from biology, engineering, chemistry, and computer sciences to design and construct new biological parts, devices, and systems. It also involves the redesign of existing natural biological systems for useful purposes. To understand synthetic biology, several fundamental concepts and terminologies are essential. 1. Biobricks. These are standardized DNA sequences of defined structure and function that can be combined with other biobricks to create larger synthetic biology constructs. Biobricks allow synthetic biologists to assemble new genetic sequences more easily and predictably. 2. Genetic circuits. These are networks of genes that work together to achieve a specific function within a cell. Similar to electronic circuits, genetic circuits can be switched on or off and have inputs and outputs, making them crucial for designing cellular functions. 3. Chassis. In synthetic biology, a chassis is a cell or organism that serves as the host for engineered genetic circuits. Common chassis include bacteria like E. coli, yeast, and more recently, mammalian cells. 4. Pathway engineering. This involves modifying existing metabolic pathways or introducing new metabolic pathways into an organism to produce a desired product. This can include the production of biofuels, pharmaceuticals, or other valuable chemicals. 5. Directed evolution. This is a method used to mimic the process of natural selection in the lab to evolve proteins or nucleic acids toward a user-defined goal. It often involves mutating genes and selecting for desired traits. 6. CRISPR, Cas9, a revolutionary gene editing technology that allows scientists to alter DNA sequences and modify gene function. It has been widely adopted for both research purposes and therapeutic applications. Lastly, biofoundries are the facilities that manage the high-throughput experimentation in synthetic biology. There are more to explore for sure. Stay tuned.